Hello everybody, thank you for joining us for the Proliferation Analysis with FCS Express webinar. Again, make sure the volume is turned up on your speakers if you're not connecting by phone. And to get started with the webinar today, we wanted to give you a little bit of background about proliferation analysis. I'm assuming most people who are here have probably already acquired some data or they're looking to acquire some data and use it within FCS Express. So the introduction of fluorescent cell staining and division tracking dyes has made it possible to monitor the number of cell divisions during proliferation and to examine the relationship between proliferation and differentiation. And software like FCS Express helps us better describe and analyzing that data using modeling tools. Now, generally, fluorescent tracking dyes can be categorized into two major groups: general protein labeling dyes, such as CFSE, which act by forming random covalent bonds with amino groups on cellular proteins, which results in a highly fluorescent membrane impermeable marker. And again, as you may know, CFSE is highly fluorescent and it's generally excited with a 488 nanometer laser. It's going to emit in the green channel somewhere around 515 nanometers. Now, there's other general membrane labeling dyes out there as well, such as PKH and cell-view dyes. These are kind of lipophilic compounds that are stably but non-covalently inserted into the plasma membrane. These general membrane labeling dyes vary in fluorescent intensity, and they can be obtained in many colors. Therefore, they're very useful for multicolor experiments because you're not just limited to using CFSE as you might do in one channel. We're going to be working with one of those dyes today for the experiment data set using a cell trace violet stain. And as a background to dye dilution and what we're doing in FCS Express and really any of the kind of modeling tools you might use is we're working under the assumption that with each cell division, when you've loaded an amount of dye into a cell or a sample, the dye is roughly divided between the two daughter cells, and therefore the dye fluorescent intensity is a measurement of the number of cell divisions that any given cell has undergone. So what you kind of see in this graphic here, we have an initial cell in that first generation. As that cell view dye, the cell trace or CFSE gets diluted out, these are going to become half as bright as the second generation and the third, and so on and so forth. And what you'll end up getting is, again, as you move from a first generation to a second, the brightness is going to be going down, and the number of cells in each generation is going to be increasing, of course. So after staining, these cell populations are analyzed at regular time intervals by flow cytometry or an endpoint. A histogram of the number of cells as a function of the dye fluorescent intensity can be generated. And typically, the cell proliferation dyes can be used to track up to 8 rounds of division before the fluorescent intensity is reduced to some sort of autofluorescence of the unstained cells. Just because you add more dye to these cells isn't necessarily a good thing. Sometimes when you add a lot of CFSE, the cells may die and you may run into other problems. But in the end, we're here today to talk about FCS Express and the proliferation tools provided. FCS Express does provide curve fitting algorithms to estimate the proliferating fractions and the generations. And this proliferation tool comes free with every professional version of FCS Express. That means when you go into the Insert tab, like you'll see here right now, you'll have the ability to insert a proliferation plot and work with that to model your proliferation data. Now, in today's webinar, we're going to take you through a sample experiment. We're going to show you how to set up some plots and gates and proliferation analysis. We're going to show you how to use a unstained control and an undivided control to set the starting and end generations for that. And we're also going to show you how to use some of the formatting tools to format those plots for publication. You may see a lot of times published data, these kind of nice looking charts, which I have up here, in which the different generations are colored in different colored histograms. So we're going to show you how to create that. And of course, how to derive all of the statistics that you're going to want to get out of these proliferation experiments, as well as how to batch process that data. We know that a lot of times when you're collecting these data files, it's not going to be, just one. You may have 100, and you need to be able to quickly report that, which we can do in FCS Express. So to get started today in learning about how to create a proliferation analysis, we're going to open up a new layout in FCS Express. So FCS Express is going to launch a new layout where we can analyze some of this data. And again, we're going to be talking about some of the more advanced tools here today. I'm going to kind of skip over some of the finer points about how to insert plots and create gates and things like that. There are a number of resources and webinars on our website for that. But again, to get started, I would like to work with these 4 data files that I have here for analysis. We have a control sample, which is an undivided sample in which we've loaded cell trace dye into. We have a stimulated control sample, a stimulated sample itself, as well as an unstained control. So we're going to be analyzing these 4 data files, and we're going to be using them to create our analysis. So to get these into FCS Express, or to make them accessible, the first thing I'm going to do is drag and drop them into my data list. This is going to load them into my data list, so I have access to them within FCS Express at any time. And since I don't have the data list pinned, I can move it and I can resize it and kind of get it to a place that's convenient for me to access. Now, I do want to start my analysis today by loading in one of my data files. It could be any. I'm going to start with my stimulated control. So I'll drag and drop that to the analysis space here in the layout, and I'm going to choose to insert a density plot. So that density plot will get inserted. I can move and I can arrange this any way that I want, just as you normally would with FCS Express. And we want to set up a series of gates that we're going to base our proliferation analysis on at first. So very simply, we will create a gate around this kind of main population here, which we're assuming to be cells and not debris. We will name that gate cells, and you'll see it gets inserted. We will also insert a gate view from the Insert tab, so we can look at and manage our gates as we're working with this analysis. Now, the next thing I want to do is open up another plot that has the cells gate applied. So I will drag and drop this gate out to the right of the original plot. And when we drop that gating position, a new plot will open that has the cells gate applied. And I'd also like to format this plot and change it from a density plot to a color dot plot. Now, what we'll do is in this experiment, you can see that we've only acquired a few parameters, but we are going to use all of them. Our Cytox advanced dye here, this is a live dead dye. The cell trace dye is, of course, our proliferation analysis, and we also have a CD4 in here as well. But we also want to come in, and with our Cytox dye, we want to create a gate around the negative population so that we're sure that when we're looking at our proliferation analysis, we are only looking at live cells. We're not including anything that's dead or dying. We're going to use this exclusion dye to remove them. So at this point, we're ready to kind of start looking at our proliferation parameter for the cell trace dye here. And I'm going to start by inserting a histogram. And this is just to give you an overview of kind of what this data looks like. If I click on the cell trace violet parameter, and then I apply my live cells gate by dragging and dropping, you'll see that this data is starting to look like the prototypical cell proliferation data, where we have some kind of peaks within our histogram. Our undivided cells are up here, and they've divided, and the cell numbers are increasing as the intensity of the cell trace violet stain is decreasing. And if we navigate between the different data files that we have in our data list, we can see we may need to adjust the gate position for one of these, right? This is our undivided control. So these are cells that were loaded with the dye, but they were not induced to divide in any way. We have our stimulated control. So this is a control where we've stimulated our sample to divide, so we can make sure that we do have divisions. And then we have a treated sample in which we really want to measure the amount of proliferation in the sample, as well as an unstained control where no dye has been loaded into the samples, and essentially we're looking at the autofluorescence of what the cell trace violet stain looks like in these samples. So when we want to start looking at a proliferation plot, now that we've reviewed this histogram very quickly, we can insert a proliferation plot from the Insert tab, and we will draw that plot. A plot will get inserted. It doesn't know which parameter is your proliferation parameter, so we just need to choose cell trace violet from the list. Now again, this is ungated, so we will apply the live cells gate to our proliferation plot, again through a drag and drop. And then lo and behold, it seems like we've done some sort of proliferation analysis. And what FCS Express is doing, again, it's using the theoretical model that you start with a fixed starting generation, you start with a starting generation, and at every kind of subsequent generation, you should find half as much dye as the original generation. And again, that's the kind of meat of this model and what it's trying to do for us when putting this all together. But again, there's a number of different things that go into modeling this data and formatting this and setting things up to get your statistics and your batch processing results. So we're going to go through them one by one. Now, very simply, when you need to derive statistics from a proliferation plot that you've inserted, you can right-click on the proliferation plot, and in the statistics category, you'll be able to insert Proliferation Fit Statistics. This will give you a standard set of descriptive statistics, such as the division index, proliferation index, the percent of cells that have divided, the peak ratio, and a number of other kind of items here that, again, are kind of standard set of descriptive statistics for any sort of proliferation experiment you may be doing. And if you right-click again, you will also be able to use proliferation population statistics. Now, these statistics differ than the standard set of kind of descriptive statistics because they are going to give you per generation results for all of the model generations that the model has found. Now, with either of these, you can right-click and you can pick to add additional columns, right? If I wanted to look at the number of original cells that a cell became, or the percentage of original cells, you can add these in there. If there's certain columns that you don't want to look at, like maybe the peak channel, you can remove them, and that will go for any of these statistics in the first statistics window that I've inserted as well. So very simply, if you're just using the default model and you're not doing anything to, kind of change the modeling or formatting of the plots, you're essentially done at this point. There's a lot more that we can do, of course. And as we navigate through these data files, you can see the difference, right? So when we go to the cell trace violet control, you can see that the model has identified that these are all undivided cells. So the division index is 0, the proliferation index is 1. And as we navigate through some of these different data files, we can kind of see how that changes and see how that updates, depending on what we're looking at. But of course, when we're working with any of this type of data, it's very useful to not just have the model run and try to identify where the starting generation is and kind of where the background generation is by default. We'd rather use controls because we have controls that we've acquired, like the unstained control, as well as the starting generation control. Now, there's ways that you can visualize where on the model by default it has set up those populations. Again, this is available through a right-click, and you can choose proliferation position markers. So if we'd like to show a marker where the undivided cells peak is, we can select that, and a line will appear and show you where the model is determining the first starting generation to be. And we can see somewhat, this is somewhat offset to the left with this data file. When we come over to this one, it might be doing a little bit better job. And of course, it's kind of blended in here with that one. But again, we're going to kind of show you some better ways to set this up. And also, if we show the background, where the model is saying that this data should end, you don't see much. It's actually kind of buried over here at 0. So by default, the model is just kind of assuming that your background is going to be 0. We don't know where it should be or where it should be set. But when we look at our controls, we can actually say, well, our unseen controls should end at about 1,000 rather than 0. So we do kind of want to modify and format this plot in a few ways to use those controls to make this behave and work a little bit more accurately. So that brings me to talking about formatting these plots and formatting them in a number of different ways. So I'm going to right-click on my proliferation plot and choose Format. And this will go for any plot in FCS Express. You can right-click and choose Format. And when you're working with a proliferation plot, you have a category here for proliferation that will allow you to change and format anything about these proliferation plots. Now, very quickly, I'm going to edit the position markers. This is something you can do very quickly. So instead of just looking at that undivided cells peak as a thin line, I increase the width. I can change the color if I want to make it maybe orange, right? You can see that it's increased the width, it's changed it to orange. It's a little bit easier to see. As well as with our background, I'm going to increase the width here, and we're going to make this blue. And what you'll notice is that on the axes here, right around 0, we have a nice thick blue line, and this is really helping us better visualize where our starting generation is and where the model thinks it should end. In this case, it's at 0. But when it comes to proliferation analysis, there is a number of fit parameters here that allow you to adjust the model parameters for your data set. Not every data set is created equal. Sometimes these things can look a little messy, especially before you've kind of optimized this assay. But there's a number of different options here for the proliferation peaks where you can change these manually. So these are the settings that the model has found by default. We always set the max number of peaks to 10. And again, as I mentioned in the beginning of the webinar, you're never going to be able to really detect more than 8 generations. At some point, you're going to dilute that dye so much that you're not going to be able to resolve that intensity any longer. So by setting this out to 10, it kind of covers all of your bases. But you can always come in and change this. You can say stop measuring or stop trying to fit after a certain number of peaks. There's also an option here to use a fixed CV. By default, the CV for each of the modeled peaks will be allowed to vary. It will use the same CV for all peaks when it's trying to fit this data. But if you would like to use a fixed CV value, or if you'd like to let the CV values kind of float for the model, you can uncheck this. And you can use those at your discretion to kind of try and make the model fit the data a little bit better. You can also see here that we have an option for used fixed peak ratio. And in a perfect world, the peak ratio should be 0.5. You're starting with a cell that has X number of intensity units associated with it, and each daughter cell should have 0.5 or half as much intensity. So this is the value that's being determined from the data. You can see it's actually 0.51, so it is very close to 0.5. But for some reason, if your peaks aren't separated as much, or they're separated kind of more, you can use a fixed peak ratio value if you'd like to try and make the model fit more appropriately. Or you could say, in addition, use the same peak ratio for all peaks. So instead of kind of letting the model float and vary between one generation and another, which sometimes helps fit the data more appropriately, you can say use the same fixed peak ratio for all values. Now, one of the really important concepts to understand with any sort of proliferation analysis in any software that you use is that these values, these defaults, are all based off of the theoretical model that one cell divides and each of the 2 daughter cells has half as much intensity or half as much staining as the original cell. And the more that you change these values, the further you're getting away from this theoretical model, and probably the further away your data is getting from being an optimized type of assay. So always keep that in mind as you adjust the parameters. If you're finding that you're having to change every single parameter in here to fit your data better in one way or another, you're getting very far away from the theoretical model, and your data is probably not approximating the model as it should be. So you can use these tools to your advantage, but then again, don't go too far outside of the constraints set by the defaults, because your data probably isn't matching up as nicely as it should be. Now, there's also options here for using a fixed starting generation and using a background generation. Now, what this means is that you can fix the point where the first peak for analysis is being set, and you can fix the background. You can say anything below a certain value or at that value is going to be the background noise. So these are actually very useful tools. They're very useful tools for the algorithm to say start at a certain point for your fixed starting generation and move outwards from there. These are not things to kind of adjust how the model is fitting inherently. It's kind of start and end points for where you want things to begin. So I'm going to show you how to set up some tools to use these values that are based on the data and based on your controls, because really we want all of our modeling based off of our controls and not just the kind of floating points with the data itself. So let's take a look at that. So again, in this analysis template right now, we are all on every single plot, we're looking at our stimulated control. But it would be nice if on some plot we could assign where the background is, because we know where our unstained control is, and if we could assign where the starting generation is based on our control. So what we're going to do is set up two histograms on our analysis template. I'm going to duplicate this histogram. I'm simply going to do a copy and paste and put it here. And what we will do is we will drag and drop the unstained control sample from the data list onto the top histogram and say replace the overlays on that plot with this data file. So now our unstained control is in our top histogram. Now we want to take our starting generation control and drag and drop that onto the histogram on the bottom and say replace the overlay on that one as well. Now what this will allow us to do is set up some markers and gates to define where our modeling should begin and where it should end. Now what we want to do for that is set up some sort of marker gate. So we'll create a marker on our unstained control, and we will create a marker on our starting generation as well. And we can use these median values for any of our kind of data sets that we want to here. But an important point to note is we're going to use this analysis template for batch processing. So eventually, I want these histograms to remain in place because there are controls, and we want everything else to vary with our samples. So we're going to do a kind of simple way. We're going to come in here and format these plots in a way that we can say for each of these plots, do not change the data file when we use next and previous or when we use batch. And again, what this is going to do is fix that plot in place and fix that data file in place on the layout so that as we vary any of our kind of iterations from the data list or we run a batch process, these values stay the same. So now that I've done that, you can see that I can click through in my data list. Everything is updating except for these two plots. Now this becomes very useful because the median for the M1 marker on the bottom plot is essentially our median that we would want to use for undivided control, and the median for the M1 marker on the above plot is the median that we'd want to use for the background control. And one of the questions that came in was just asking if I could kind of repeat those steps, essentially how to kind of fix these plots in position. So again, keep in mind, if you ever need to fix a plot on your analysis to a data file, whether it be for proliferation analysis or an isotype control, really whatever it is that you want to do, you can right-click on a plot and choose Format. And in the Overlays category, you'll have the option to change data file during next, previous, and batch. That is the default, and you would simply uncheck that. You can also do that by multiple selecting plots And in the Format tab, you would access the overlays here, and again, change that on all of the plots that you've selected at the same time. So again, this is one of those functionalities that's there for any sort of plot that you use within FCS Express. But we're really just about there now and completing our analysis based on our controls. What we want to do for this proliferation plot now is format it so that we use the median value for our positive control and the median value for our unstained control as these starting and end generations. So again, what we will do is format this proliferation plot. And in the proliferation fit parameter options, instead of using a default fixed starting generation value derived from the data, we will delete that and click on this little T value for token. Now, what tokens will allow you to do in FCS Express is choose any sort of value or statistic or something in the software that may be there. So we're going to choose a statistic, and we can pick our non-dividing control plot, which is that histogram that we fixed down here. And the statistic that we'd want to use is the M1 marker and the median. So what will happen now is that when we choose this value, and I'm going to choose to display just the value in this token, you can see that this value is highlighted in gray, and it will use that fixed starting generation as defined as the median from the marker in M1. And you may have seen that that marker just kind of shifted around a little bit on the proliferation plot, but if we move that marker, you will see that it will update for us on the proliferation plot, and all of the statistics associated with the modeling will also update for us in real time. So that way, if we find that there's a few outliers here and we want to kind of tighten this up to make sure that we have a more accurate representation about where this proliferation analysis modeling should start, we can do so very quickly and easily. Now we want to do the same thing for the background generation. So we will format this plot again. We'll come into the fit parameters, and we will use background noise, and we will assign that to a statistic. And again, that statistic is going to be for our histogram for our unstained control. This is going to be the M1 marker and the median for the M1 marker. Now, you will see a difference. We will change this to the value, right? So instead of using 0, it's using about 1500. And you can see that this marker has now kind of jumped up to this position. That's the median of the value from our M1 marker. And again, if we were to adjust this marker, you can see that that change in that value is updating for us in real time, and it will be impacting the fit statistics for any of the data that we're looking at down here as well. Now, with those fixed, as we move between data files, you can see that those marker positions have not moved. They're all based off of our positive and negative controls. And in this case, we have 2 samples, but if we had 200 samples or 2,000 samples, it would remain the same based on our controls for all of our samples all the time. And this is really one of the most appropriate ways that you can be fitting this data, is basing where the modeling should start off of your off of your starting generation control, and where that modeling should end based on the autofluorescence of your unstained control. So at this point, we're really done with our analysis. We've fitted this data. We've done it appropriately based off of our positive and our negative controls. We've reviewed our gates, we've reviewed our samples, but there might still be more we want to do, because if we want to batch process, say through 1,000 samples, and we want to generate some sort of publication quality figures, we'd probably want to format these plots a little bit more to make them nicer. And one of the things that you see in publications quite often is proliferation plots that look like this with different colored background generations that are in there. I think this is a very striking way to represent the data, either for publication or even just at lab meetings and things like that. So I'm going to show you how you can format these proliferation plots. Again, it's in that right-click and format. You can see we keep kind of going back to that to get to the tools that we need to change things about this plot. And when you click on overlays, you will see that we have every overlay on that plot, all the generations and the undivided cells, they're all listed as different overlays which you can assign different fills and fill styles and colors, using any of the options that are here. So a very kind of common way to do this is, select all of your generations, and at a very basic level, change from our default kind of hatch marking to a solid fill, and put in some sort of transparency associated with that. And you'll see that when we preview this chart, it now looks much nicer. We're using the same color. We're using, instead of hatch marking, we're using a solid fill with a transparency where we can see the overlaps. But in that same vein, we can come through, and in every generation, we can assign a different color. So for generation 3, we'll assign some sort of violet. Generation 4, we'll go to blue. I'm just going to kind of keep going through here using the color palette to, assign different colors for all of these generations. And what will happen in the end is when I go back to the plot or when I click preview, you will see that we've assigned different colors to all the different proliferation generations in here. It looks like I've assigned some sort of almost like a pastel here with the kind of random color palette I chose. But again, you can come through and you can change these to any sort of color palette that you want, any way that you figure is the kind of best way to represent this data and these publication ready figures. You can go through and change that, again, any way that you need. So at this point, we have more than just an analysis. We have a presentation. We have a presentation-ready figure. We have an analysis, and we have some results here, all based on our controls. Now, when you're ready to kind of batch process this data and work through all of your controls very quickly and easily, we can set up what's called a batch process in FCS Express, where we can export all of the results that you want, any of the figures, anything that's in this layout, any way that you want. And it's very useful for working with proliferation data. So what we will do is set up some batch actions. Now, the first action I'm going to set up is I'm going to ask FCS Express to save all of my samples to PowerPoint. I'm going to say export them at a very high resolution, very high quality, and I'm going to say that when these files get saved, I would like them saved to my desktop. And on my other screen, I'm just typing this in. What you'll see here, as I've said, to save this as a PowerPoint on our desktop. And what it will do is, in this case, we've asked FCS Express by default to put everything into the same PowerPoint presentation. So when this batch process runs, it will start from the data file that happens to be on the data list, and it will export to PowerPoint whatever is on this page with each new slide in PowerPoint representing a new data file. Now, we also have a number of statistics here that are interesting for us: the division index, proliferation index, the per generation values, and these are probably better suited to be exported to Excel directly rather than being exported, one by one through a drag and drop. Now, I do have somebody who just wrote in and said they lost the audio. I just wanted to confirm that everybody else can hear us. If everybody could just type in a quick hello and let me know if you can still hear the audio. It might have just been that one user who got disconnected. All right, great. So I think one person just lost audio, so you'll have to try and reconnect there. So again, what we're going to be following up with here is trying to export directly to Microsoft Excel. So what we will do is choose Export to Excel column mode. And we will tell FCS Express that we want to save this as an Excel export. We want to save it on my desktop. I'm on my other screen here. I'm typing this in, and you'll see that we're going to create an Excel file on our desktop for this analysis. But we still haven't told FCS Express what we want to populate into that Excel report. So what we will do is simply start by dragging and dropping some of the information that we want into that Excel report. Now, at a very basic level, we want reported for every data file the file name. And we can derive that from the plot. So we can simply drag and drop this whole plot into there. We're going to choose statistic token. And in the statistics category, there's an option here to choose file name. And when we do that, the first column in our export to Excel will include the file name. Now, very simply, we probably want to report all of our division statistics here. So we can quickly just highlight everything that's in this table and drag it and drop it into our Excel export. And it will all be populated into our Excel export on a per data file basis. And again, if we wanted to come in and say for all of our generations, show us the number of original cells, we could grab everything that's in this list and we can drag it and drop it to our Excel export, and it's going to start populating that back into our Excel report when we export that. So you can see just through a few drag and drops here, we've actually populated an Excel report with a large number of statistics. There was really nothing we had to do there, and FCS Express is going to report all of this for us essentially with one click. So I'm going to close this batch process action window here. We really don't need it anymore. We've told FCS Express to export to PowerPoint and export to Excel. And what we will do is we will change to the data list where we want our batch process to start, which is going to be iteration number 1, and we will click Run on the batch process. And what you'll see is that FCS Express is going to start iterating through all the data files in our data list. It's doing two of two things. One is exporting to Excel and one is exporting to PowerPoint. And it's doing that on every iteration or data file that's in the data list. We are not varying what's in these histograms because those are our controls. And Excel and PowerPoint are being launched for us, so we can review this data immediately. And what you'll see is that in the first PowerPoint slide here, we have our undivided control. Our second PowerPoint slide, we have one of our samples. The third, we have another sample. And the fourth, we have our unstained control. And again, when you have these out in PowerPoint, they're all high resolution vector graphics. You can make these as big as you want. You can move them, you can resize them, you can group them, you can ungroup them. And really, this is your publication ready figure. Right? When you're ready to publish or do a lab report, you simply have this all out in PowerPoint. But again, let's review what's come out to Excel as well. We've told Excel to export our file name, right? We never had to enter this in. It's all derived from the data file and the plots itself. We have all of our division index statistics, our proliferation index statistics. They're all collated for all of the samples. And if you remember, our unstained control or our starting generation control is at the top, our unstained control is in the bottom, and of course this data makes a lot of sense. And when we start looking into the statistics for each proliferation generation here, here's the undivided cells. We're looking at how many cells are in the undivided generation, generation 1, generation 2, generation 3. And these results make a lot of sense because in the second two samples, these are our samples, and these are our controls in the first two rows. So you can imagine if you had 200 samples, this batch process would run over 200 samples, and you would have 200 rows in Excel telling you, all of your division index statistics, all of your sample names, your file names. If you had well identifiers in there, would all kind of populate and appear. So again, I do really recommend using the batch process to review any of this data or to report out, if you're working with even just a few samples, it's very quick and easy to do that. Now we should mention that, you can work with these proliferation plots just like any other plot in FCS Express. I know another kind of common way of looking at proliferation data is to insert some sort of density plot and look at, one parameter versus your proliferation parameter. We'll apply the gate. We can kind of see where these division breaks are there in the density plot. And you can create any sort of gate you want. If you want to create a gate on this plot, you can simply do so. The gate will get created, and you can even apply that back to the proliferation plot to kind of just look at the amount of proliferation going on in those generations. And, conversely, you can create a marker on your proliferation plot. You could simply right-click and convert that marker to a gate. And what that will allow you to do is apply a gated population, like this M1 gate, to another plot, and only look at the generations that are appearing within that gate. So we'll get this applied to the plot over here, and you'll see that when we look at this, we can easily assess in any sort of other density plot kind of the information going on with that. And of course, this is very helpful when you're looking at your proliferation parameters versus something else like another marker, in this case CD4, and we can look at high expressors of CD4 versus low expressors of CD4. And you can imagine if you wanted to model each of these separately, you could set a gate on the high expressors, set a gate on the low expressors, and apply them over to two separate proliferation plots to analyze these things. So again, what we've covered today in just about half an hour or 45 minutes is essentially, how the modeling is used. And when it comes down to it, proliferation plots are inserted, formatted, and used just like any other plot in FCS Express. So, you don't have to consider them any different. They do have specific formatting options to allow you to change the fit parameters as needed. Again, keep in mind that the further you get away from the theoretical modeling, the kind of worse your data might be. And in some cases, you might want to go back and reacquire or restain and reanalyze that data. Again, the further you get away from that perfect model, the kind of the worse the data is. Now, I do recommend using the starting and background generations and basing them off of controls. We make this very kind of easy to do in FCS Express. If you remember, what we did was we inserted two histograms, one for the positive and one for the negative control, and we assigned the fit parameters for the starting generation and the background noise based off of the median of those samples. And again, batch processing is going to allow you to quickly analyze any number of samples. In this case, we just looked at 2 samples and 2 controls, but it's very easy to extrapolate and scale up to 20 samples, 200, 20, 2,000, it doesn't matter. Now, outside of the webinars, we do have a number of tutorials that cover the basics, that cover formatting, they cover batch processing. So if you're more of a kind of written learner, you can come in to the FCS Express manual, you can come into the tutorials, you can access these from our website, you can access them from within FCS Express, and you'll find that in our tutorials here, there's a tutorial for proliferation analysis. You'll be provided with the sample data sets that we use today, and it's going to bring you through inserting proliferation plots, formatting proliferating plots, as well as batch processing data using the controls that we described today as well. Now, additionally, in the FCS Express manual, there's a number of kind of resources there to kind of help you better understand what the fit proliferation statistics are, and kind of how they're calculated and all of the formulas, again, how these things are actually derived. So I do recommend, if you have any questions about the mathematics involved for the modeling and how any of these things like proliferation and division index are calculated, you can access this in the manual. When you access the manual on the website or within FCS Express, you can simply search on that manual for proliferation, and it should bring any of that up. So again, I hope everybody has been able to learn a little bit more about proliferation analysis today. I saw a number of questions come through that we'll kind of talk about in a minute. But if you do have any questions going forward, you can always email us at support@denovosoftware.com. We'll be happy to answer your questions there about proliferation analysis or really any sort of analysis you might be doing. If you're not currently using FCS Express, or if you have colleagues that you think may be interested in using these tools, you can always go to the website and download a free trial of the software, which again is going to be fully functional. It's going to include all the proliferation analysis tools. So again, we do recommend going there and getting that if need be. So again, thanks everybody for joining us today, and we'll be following up with questions in just a few minutes.